So good morning, everyone. Thank you for taking the time to join our webinar today on asbestos and also, asbestos in general and volume measurements specifically across multiple areas, multiple determinations. My name is Neil Shepherd. I'm the Sector Manager for Life Sciences, and it's my pleasure to host this webinar today. A couple of rules. We're going to record this session and we will use some of the information to make available via our website for those who couldn't make it today. Questions, we will hold off on the questions. I've only got a couple of slides. So this is not going to be death by PowerPoint. So I've got a couple of slides and then some discussion around that. And then we can have some questions if you need to ask questions. I'll just say that this is not a consultation. We have already published documentation around this. This is more by way of an explanation, and it's an unusual thing for me to do. Normally we would publish the document and get on with the assessment activity, but we have had some feedback, and I thought it was worthwhile to go over the changes and make sure everyone is on the same page. I mean, this is a team effort. We are the assessment body. You are either technical assessors, NATA, my colleagues at NATA who do the assessments, and or regulators or consultants. So we all have some skin in the game here, and it's just an opportunity for everyone to sort of come together and listen to what I say. So you all hear it once. You can refer to the documentation. You will have read the documentation, both the Life Sciences Appendix and the specific accreditation criteria for asbestos. So they've both been published for a while now. And I think we can go from there. I'm going to share the screen. So just with questions, I think you can put questions in the chat and we'll hold the chat until the end. Any questions that I do not have the answer to right now, I'll take them on notice. And we can have some verbal discussion as well if anybody wants to actually ask a question sort of in real time. So I'll just share my screen, and as usual, dangerously close to the leave button, but I've managed to get right on everything. So the first, hopefully, can you all see? I can't see you now, but you can see my screen. Oh, it'll come up on the other screen. Excellent. So the first thing I'm going to talk about is just the process we use at NATA when we review an outward-facing document. And when I mean an outward-facing document, that's one of our accreditation criteria documents. So those are the documents you can download from our website, and they include both specific and general accreditation criteria, guidelines, and other information. So in this instance, I'm talking about the process we followed for both the Life Science Appendix and the Supplementary Accreditation Criteria for Asbestos, the SAC as it's called. So this is just the process here. So we start with a review. Now we do these reviews periodically, maybe every 2 to 3 years, or maybe a bit longer, depending on the nature of the document and how fluid the technology is that the assessment activity is covering. So if nothing's changed, then we might review it and it might not have any changes in it. And sometimes we might feel that industries change, technologies change, and we need to update the document to keep up to date with progress. So the process starts with a review, an internal review with advice from the Accreditation Advisory Committee. So they're providing us technical input into the content. Once we're satisfied with that draft, we do a final internal review. And then the document is published by Anne-Marie in most instances. It goes out on, there's a notification that the document is available from the website, and you can look at that document and review it. And if you have comments on that document, questions, etc., then there is a form that you can complete. So that process takes a month. You are notified that the document will be available for public comment for a month, and you can submit the commenting table during that period. Once that commenting period is finished, the document is removed from the commenting period finishes, and myself and my colleagues review the comments received in a table. Now, because they come from many and varied stakeholders, we have to sort the comments, and we sort the comments in the table into the clauses in the standard. You are required as part of the commenting to not just make a comment. If you want to make you need to make a suggestion for a change if there needs to be one. So we do have some rules around that. If you just sort of say, I don't like this, well, I don't know what to do about that other than if you don't like something, you need to explain the reason why and suggest maybe a change. We review all of those comments, we sort them in the table, and we make a decision along with the Accreditation Advisory Committee, subject matter experts, whether or not a change is needed, and the document is ultimately published. So there are three things. You may get no feedback. I have had the odd document with no feedback. I've had documents where I've had to do more than one public comment stage, and the process just follows through until we're satisfied that the document is satisfactory to be published, and that's the end of it. It goes out for publication. When the document is published, it gets a publication date and an implementation date. And because we realise that assessments are going on an ongoing basis, it wouldn't be fair if you received a new document and were assessed against it the next day. So we give everyone there's a 3-month period within which to sort of, if you need to make some changes to the way you operate your business to comply with the accreditation criteria, then that's what we ask you to do. So you've got 3 months. It might turn out that you've only got 4 or 5 weeks because you get your document, you have your assessment straight after the 3-month period. Some people might have another 18 months. But it's luck of the draw. There is very little we can do about that. But my observation is that really, when that document is published, regardless of the 3 months, you should look at what the changes are, and you should start an implementation plan in order to be ready for when your assessment activities occurs. That is all I have to say on that. Hopefully that's clear. So I'll stop sharing that document. And I'll move on to, I couldn't decide whether to do the volume measurement first or the asbestos. I think I'll do the volume measurement first because that covers more defined. It sounds useful. I'll do the volume measurement first because that covers more than just asbestos. So I'm going to share the screen again. Just checking everyone can hear me. You can share the screen yet. So this is the specific accreditation criteria document for life sciences, the appendix. I'm not going to go through the whole document. I'm only really talking today about volume measurement. You can see I've highlighted some sections in the document. So it's really from page 11 through to page, I think it's about, yeah, it's at the end of the document. So from page 11 onwards, this is what we're going to be talking to today. So there has always been a requirement in the asbestos SAC in relation to volume measurement and quantification of results. It has always been a requirement that where a facility, where a conformity assessment body is in control of the sampling process, they put the pump out, they know the pump's functioning correctly, they do all the relevant checks, they apply the filter, they take the filter and send it back to the laboratory, etc. Then you can, because you're in control of that volume measurement, you know the functioning of the pump and you know the volume of air that was passed through the filter, you can issue a quantitative result based on the number of fibres counted per metre squared. Now, when you can't do that, there are a number of circumstances. So filters arrive at your facility from a third party, or there's another circumstance where an unaccredited facility or some other organisation sends you filters, then you can only issue the results as fibres per field. You can't, because you do not know what the volume of air passing through the pump was. And there's a critical thing there, that the volume is used to calculate the concentration. So that's always been the case with asbestos. Surprisingly, and I am surprised about this, I don't understand why facilities doing other testing such as mould testing or crystalline silica or dust using a very similar process would not also have considered that needed to be done. We don't have specific criteria documents in those areas, and because really the the only reason we have it for asbestos is there are a number of issues, other issues related to asbestos. But that process, that requirement for understanding and controlling the sampling activity in order to know the volume that went through the filter applies to all of those types of activities where a pump draws air through some sort of filter or membrane or something, and the target determination is caught by the filter, and the air passes through. Of course, you never have the air. So what am I trying to say? So the volume is critical in all of those areas. So as you can see, I've been quite explicit here in saying, you know, this covers asbestos, mould, silica, and dust, and any other analysis, because maybe one day something else will come along that I haven't anticipated. But when you use a pump type apparatus in order to collect the determination on a filter or other substrate, then this criteria applies. If we look at the reporting concentrations, it's quite straightforward, really. You can report fibres per mil, spores per meter cubed, milligrams per meter cubed of whatever substance in the following circumstances. The facility is accredited for the relevant determination concentration and has collected and analysed the samples. The facility is accredited for the relevant determination concentration and receives samples from another accredited facility that can do the volume measurement, and we've assessed it. Or the facility is accredited for the relevant determination concentration and receives samples from an external party that is appropriately supervised. Now, I very rarely see a facility who is able to meet that criteria. Supervision is more than training some operatives to go out into the field and operate and operate pumps and send filters through. In order to meet that third dot point, you really have to be actively supervising the operators in the field. I won't call them third parties. We stopped using that reference a good many years ago. So when I mean supervise, I mean you know what they're doing. You're the testing facility. They're working on your behalf. There's a partnership there. You know where they are. You know what they're doing. They've discussed it with you. You know that the equipment they're using is calibrated. You may have calibrated for them. They may have got it calibrated elsewhere, but you have access to those records. You will know where they're putting those pumps, and you have an assurance that the traceability is there when you receive those filters. Now, it's not an easy thing to do. I think that's probably the hardest of the three, because what tends to happen is there's some initial training, and then the operatives in the field go off and do stuff, and there doesn't appear to be that much supervision. So I'm cautious about that one. It's available, but it is not an easy thing to demonstrate on an ongoing basis that you are managing that appropriately. But by all means, that option is there. The other point there in relation to reporting from external parties is just a summary of the way you would report the information. So asbestos fibres counted on Y fields, mould spores counted on traverses or percentage of deposit, and the RCS dust as a percentage of mass collected. So there's no. So really that in a nutshell, that's what I wanted to talk about in relation to volume measurement. So the ultimate thing there is that it applies to any activity where air is drawn through a filter, and the determination is made at the facility. The concentration is calculated at the facility. You must have assurance of the traceability and the appropriate and fit for purpose activity of the sampling equipment and the operatives doing the sampling. That would, of course, occur if you were sending your own staff out. You would know they had their own equipment. They would go out to a particular work site to do a particular job, which had been discussed. You'd know where the pumps were being sited. You would know, you would have records of the calibrations of the pumps. You would have records of the field checks that are being undertaken, and you would be able to substantiate all of that by being able to provide us with those records or evidence of those records during an assessment activity. When you don't have that, then you are in the space of issuing results as a qualitative result. I don't know if we've had any questions, Anne-Marie. Have we had any questions yet?
David Honey has asked a question in the chat. Did you want me to read it, or you want to read it?
Because I'm going to move on. I'll move on to the field site. So maybe we'll do these first. Otherwise, we'll sort of get ourselves mixed up.
Okay. Well, David's asked, does supervision of an external party mean being there every time? Some labs train external operators, and we have accepted that as a valid volume measurement. Otherwise, is there a mandated frequency of supervision?
There is not. Thanks for the question, David. There is not a mandated frequency of supervision, but I would say you as an organisation have to establish a process that gives the accreditation body NATA confidence that the supervision is occurring on an ongoing basis. To me, supervision is in multiple parts. There's making sure the staff are competent in what they do, occasionally visiting them to make sure that they know what they're doing. You might want to audit them on site. You may want to observe them. I mean, it's possible these days with this type of technology we're using here to get them to watch what they're doing in the field. You don't necessarily have to be there. So there are multiple ways in which you might achieve supervision, but at some point the question was, do you have to be there all the time? The question is no, not all the time. So you wouldn't supervise the staff on your own. You wouldn't stand over the staff in your own laboratory all the time. You would periodically implement some sort of checking to make sure that they were doing what they're supposed to be doing. And also, I suppose, instill in them the understanding that they should be asking you if they're unsure of what they're doing. If if something unfamiliar happens, then rather than proceed, that they refer back to the facility, the supervising organisation, and they check before they proceed. Any more questions?
All right. Neil, Linda Apthorpe has asked, guessing then if that occurs, it would also require establishment and following a sample plan for the third party. Would that assist with the supervision?
Well, thanks, Linda. Well, you could. I mean, we do not dictate exactly how these things are done, but there is no reason why you couldn't say, well, we are going to visit. We've got three I'm going to use a term third party. We have three external operators in the field. They work 2 weeks out of 4. I'm going to periodically visit them, say, once every 6 weeks or something. I don't know. I'm just making it up. But having a plan and following a plan and having the evidence you follow the plan is always good when you're trying to confirm with the accreditation body that you are meeting the requirements.
And Bob Simons has said, volume measurement, does this apply to dust deposition gauges? Australian Standard 3810.10.1.
I'd have to take that on. If it's I don't actually know how a deposition gauge works. Is it drawing air or is it just sitting there and something lands on it?
You can open Bob's microphone to confirm. Oh yeah, Bob said just sitting there.
Yeah. Well, you're not. So the process there, thinking it through, sitting here, is that you will want to know it was there for the appropriate amount of time and in the right place, because so it's a gravimetric test at the end of the day, is it? You're looking to see what the filter weighs before and after the period of exposure. Bob, hear that?
Go ahead, Bob. I've just opened your microphone.
Sorry, Neil. It's a 4 or 3.8 litre bottle with a funnel in it that's set out for 30 days. So there's no volume.
There's no volume. So it's not a volume measurement in the same way. So it would be like, so when you receive the sample, you'd receive the whole sample in the laboratory and analyze it as you would like water or something like that.
Correct.
Yeah. So it's not quite the same thing, but I would suggest, as with everything, you either have samples as received for that, or you want to know. I mean, is it going to make any difference the level of supervision you give to whoever's putting that piece of equipment out?
No, it's the same as sampling soil or sampling groundwater or sea or marine water.
So in that instance, I would say that it's not the same as what I'm talking about with volume measurement.
Thank you, Neil.
I'm particularly focusing on the functioning of the pump and knowing, having an assurance that when you calculate the volume, knowing the speed of the pump and the amount of time it ran for, that if you can't have that assurance, then you can't do the quantitative, and you can't do the quantitative measurement. You can't report the quantitative result. Any more?
Stuart, Kia, is there any requirements to do back pressure checks on sample pumps?
Yes. It is one of the normal functioning activities. I'm not a technical assessor, by the way, but back pressure is one of the things you would be doing in order to make sure you are using the appropriate pump and the appropriate filter size in the field. There's a hand from Ibrahim. Bit tricky with 8 screens, isn't it? You're doing very well.
Yes, sorry, switching between. Okay, go ahead, Ibrahim.
He's on mute. Is he typing? Oh, no.
Yes, I've done. Okay, hang on.
Maybe type it in the chat, Ibrahim.
Yeah, sorry, it doesn't seem to want to. Allowed your mic. You can unmute yourself, Ibrahim, if you can try.
Right, well, maybe if you can try type something in the chat, we can move on. There seems to be some technical glitch there.
Neil, there's a few other questions in the chat. Do you want me to read them out, or do you want to go into the chat yourself and have a read and answer as you go? It might be quicker if you read them.
Huge. Yeah, Karen.
Next one, Powell Kidden is next. Will supervision requirements be communicated to technical assessors? In previous roles, I was informed that supervision would require video calls to be made with staff setting up pumps in the field if they were doing it under the form of regulations. So I'm hoping there are a number of technical assessors on this call. We did provide the communication to all facilities, to regulators, and to technical assessors. But our lead assessors are very aware of this process, and as they are leading the technical assessments, they will be able to devise prior to each and every assessment the requirements around being able to ascertain whether the supervision is being done correctly. But yes, as I said in my earlier comments, one of the ways you could supervise external parties in the field is to use some sort of video or sort of remote surveillance activity. But I would still suggest that on occasion you would want to be in the field, because there's one thing to watch it through a camera, there's another thing to be there actually observing what's going on, because it allows far more questioning. And really what you want to do with your supervision, I mean, the whole point of supervision is you make sure that the staff you are supervising are doing what you want them to do. So Kira Bailey, does anyone have any idea on how to apply this, including back pressure to mould pumps? I will have to let
I don't think we can answer that question right now, but if anybody wants to communicate with Kira about that, we can take that on notice. No. So Patrick's answered. Yeah.
Neil Ibrahim is unmuted now. Ibrahim, you can go ahead and ask your question.
Yeah. Yeah. Can you hear me now?
Yes.
Yes. Welcome.
Yeah. Okay. Good. Okay. Yeah. So I have one question here. We have been doing the mold analysis for some time now and reporting the results in spores per sample and spores per cubic meter for the volume with the volume measurement, but we're noting that the volume measurement is not covered by NATA accreditation, clearly stating this on the report. Is that still acceptable, or it cannot be included at all on the report?
I think that I see what you're doing, but I think the issue is that in order to do that, you're really sort of you're still doing something that's not necessarily technically valid because you can't prove the traceability. So you can't do it. It's not quite the same as let me think of an example. Water. So you might sample water and is this going to be a good analogy? You might sample water and somebody sends you a pot of 500 mils of water, and you take 100 mils from it, and you can measure that 100 mils. You know that in your laboratory you measured that 100 mils. Whatever happens in this mold space, because if you're not in control of the sampling activity as I've described, then you can't assure the volume. So you've got no way of demonstrating that your quantitative result is actually correct. So just sort of saying, but it's not covered by the scope of accreditation doesn't really fly, doesn't really count, but it is not appropriate, because ultimately the reader of that report is gonna look at that and go, well, I've got 65 multiples per centimeter squared or something. So you can't do it.
Okay, so the client has to do the calculation based on what they're going to do.
It's the thing, isn't it? I mean, the client, you can have any arrangement that you like with your client, but what we're saying is that our obligation is to you is our arrangement with you is what we can actually assess, and we can only assess the activities you do.
Yeah, understand.
I think it also needs to be clear with the clients and maybe the regulator, anybody else who might be looking at these results, is that there isn't an assurance of the volume. The one way around all of this would be to have a lot more accredited sampling. Now, I'm not trying to sell accredited sampling. What I'm saying is there's a gap in the market here. There's a lack of sampling activities covered by accreditation.
Sure. Well, the majority of the air sampling pumps for the mold specifically, they cannot be calibrated and obtain a volume measurement to ISO 17025. There are a couple of exemptions, but the majority of what's being used in the industry, they cannot be. They don't have back pressure and cannot be calibrated to the accuracy. So that needs to be probably made clear to all clients as well.
That's the thing, isn't it? You should be using equipment, or not you particularly, but conformity assessment bodies should be using equipment that's fit for purpose. And if you're setting out to do a volume, a concentration, a reported concentration, then you need to use equipment that you can generate a result that you can assure the volume of. So sounds to me like some of this equipment is not fit for purpose.
Yeah, sure. Okay, thank you.
Okay, thank you. Is there any more questions? David again, mate. Nancy. Lab supervision to a third party can do the sampling activities. This is on condition that the laboratory holds accreditation for some. Nancy, so you could use another accredited facility that does the sampling, that would solve all your problems, wouldn't it? Because you would know that that facility had met the requirements for accreditation for sampling. If that's the question you're asking, do you want to confirm? No issues on the 8th screen. Well, if she responds, I'll move on to that. So, David, if the sampling is non-NATA accredited and you want to report a concentration, you do issue a separate report that does not include the NATA logo, and you should make it clear. Yeah, even then, David, I think the issue is that you haven't got an assurance of the volume. I'm saying that this isn't a sort of a thing saying, well, this is NATA, not NATA. This is saying the fundamental problem is if you don't have control of that sampling process, then you don't have an assurance of the volume. So technically, can you really substantiate that the concentration that you are generating through that calculation activity is actually correct? And I don't think you can. So I don't think issuing another report, it's a bit like issuing not putting an endorsement on, or saying this isn't covered. You just can't do it in this circumstance, unless you have an assurance of the volume through the traceability chain. Are there any more questions? Thank you. I appreciate the questions. It was good. I hope I've answered them all. What I can do is I can move on to the other document, which right. Okay, so hopefully that's big enough. So there are a couple of things here. So field sites. Field sites are a peculiar thing to asbestos. I didn't invent field sites. I inherited them from a previous field manager at NATA. The idea of a field site is that to the advantage of the customer and the conformity assessment body, you locate some of your equipment near to where the work is being done, and that's always a question of how near, but I'd say very near, very, not 5 kilometers away. You're putting it on the site of where the work's being done in a location where it's appropriate. So you've got a porter cabin or something else. We have had issues in the past with hotel rooms and caravan sites and things like that. I think in the previous version of the supplementary requirements for accreditation, we said you can't do that. I don't believe any hotel owner or any caravan park owner or anybody else would appreciate their hotel rooms being used for business practices, laboratory type practices. And if you use the word asbestos, then you might be asked to leave. And NATA really, we are very strongly saying to you that it's not appropriate. You need to use for a field site a purposely set up area in a porter cabin or some other thing that has the appropriate equipment and the appropriate accommodation requirements to meet as you would to do the testing in your laboratory. This is an advantage to the customer and to the conformity assessment body because it's making the return of results quicker because you're right on site. You're not having to transfer them. There's another thing here. We do on occasion get asked the question as to whether or not ad hoc. So a fire occurs or some sort of disaster. Someone drives their car through a shop, or someone gets up on the roof of a warehouse and pressure washes it or something like that, and you're called out to go and check what's going on. That's not really a field site. That's an ad hoc sampling activity that's occurred. You haven't planned. I mean, I think the difference between ad hoc and field site is one's a planned activity, and one is there's an emergency situation, there's a fire or something, and you're called out by the council to say, is there any asbestos here? You take your equipment, you go and do some analysis. The thing about ad hoc sampling is that you need to make sure that when you take your equipment, you transport it in an appropriate manner, and when it gets there, you do whatever checks you would normally do to make sure it's functioning correctly. So, I mean, that's really all those couple of paragraphs there. So when we talk about field sites, we have made some changes to the field site requirements. The field sites now can operate for 12 months. I think previously it was 18 months. So there's a 12-month period. I don't know how many operate for more than 12 months, but that is, it is important to make sure that you don't run past that. I mean, we'll talk to that a bit more to that in a minute. But the big change is that rather than us ask you prior to an assessment activity whether or not you have any field sites, we are introducing a process where you need to notify us. So we will have a form available for the notification of establishment of field sites, and all conformity assessment bodies who are in the business of operating field sites will need to notify us that they, when they open one and when they close one. But I'll start with the beginning of the process. Is that so? All facilities that have one will have a process of how to establish them. There might be many and varied different ways. I mean, we don't describe what you need to do and what type of equipment you might use or what type of accommodation you would think is suitable. That's your business decision. But ultimately, you will have a process that we need to review and say, yes, that is acceptable. You're not using hotel rooms. You're not doing this. You're not doing that, etc. So there is a process of asking for approval to be able to set up field sites. And once you get that, then you're on your own recognizance. You can set up as many as you like. The one thing you need to do is each time you set one up is to tell us. And our staff will respond to you and say, well, yes, we've kept that on, we put that one on the list. And ultimately, what that will help with is when we're planning our assessment activities, we'll know how many you've got and where they are, and we can plan to visit some of them. And so prior to the assessment activity occurring, the discussion will occur between the lead assessor and the and I see Jade has a question. The lead assessor and the authorised rep as to where these sites are, the number of them, how we might access them, or some of them, and we will sample a number of them depending on the number you've got. Jade has a question. Might as well do the questions as we're going along.
Go ahead, Joe. You can unmute yourself.
Thank you. Hi, everybody. Just quickly, when you're talking about the ad hoc one-off situations, look, I understand that's actually kind of great, but at the same time, if there is a fire, you know, it does say one-off sampling or testing activity. So if there's a fire, it might take a few days in order for that to actually be dealt with and to be handled. How do we know how long an ad hoc situation actually is before it becomes a job, if you know what I mean? Because I understand that it's like reactive and we're jumping on it straight away, but the scope can change over time. Like, what kind of a time period are we looking at there before we start to consider that an actual job that we've taken on and not just a reactive work?
It's one of those how long's a piece of string thing, isn't it? But I would say that, I mean, if you say, oh, there's a hospital with 9 floors and it's being refurbished floor by floor, and it's going to take 6 months or something, and I'm going to put my portico in there, that's clearly a field site. If someone phones you up and says, oh, there's been a fire at this factory and we think there might have been asbestos, and you take 3 days to go around there examining stuff, I'd say that still fell under the ad hoc. That's how, I mean, I don't, if it went on for months though, I mean, you know, how long would it normally take you to determine if there was asbestos contamination after a fire? Is it, I mean, once you find it, you find it.
Yeah, like generally straight away.
It might morph into something more, in which case you might have to take the field site. So I think you really have to sort of take it on a case by case basis there. But you can't. I don't think we'd be particularly impressed if we turned up and this ad hoc was being used for something that had been going on for weeks and weeks. And clearly, it was a planned activity.
Yeah, that's fine.
This is a partnership between us, the testing of, you know, the conformity assessment bodies. We're trying to establish a clear pattern of a distinction between these two things and make sure that we're all aware of what's going on. And so when we do our assessment activities, we can plan and you can know we're coming and you can have the information, you know, the records ready for us. And it should make life a bit simpler, I would hope. Yes, you do have to fill a form in, but you know, there's no way else of us knowing, is there really, other than you complete that form. So, what else can I say about these things? So
Neil, Bob has a question, if that's all right.
Sorry, I think the recording thing is over.
Yeah, go ahead, Bob.
Thanks, Anne-Marie. Neil, we have an accredited mobile laboratory. How does it fit?
Okay. So the mobile facility would not be a field site. It would be, it's associated with whichever one of your sites it operates out of. And so what we would do is we basically look at that as like another room on wheels. And so when we do those assessments, we would ask that the mobile facility would appear in the car park of the building or something. I mean, we could actually travel to where it's being used, but I would suggest it's probably no more worthwhile doing that than it is to just sort of get it to come to one of your static sites.
Thanks, Neil.
Okay. All right, I'll move on. So there's a couple of things we need to know when the field sites. The process is, the first thing is we're going to have to approve everyone for having field sites, and we can do that. The option to run field sites if you're doing it, and then if you've got any, you'll need to tell us where they are now, because some of you will have them operating and when they started, when they're planning to finish. And then as you close them or open new ones, then you just communicate with NATA, with your client coordinator, and they will record that information, and that information will be used as assessments are set up going forwards. What else can I say? The other thing is, I suppose, is what we would try to do is assess the field sites in conjunction with one of your surveillance activities. So periodically, we will visit your organisation, and we will, communicate with you in advance of that is happening. If you've got field sites that can be assessed at the same time, then we will try to do the whole thing together. It probably means we'll have to, we might have to go off site, we might have to go away, travel away from the actual static site, which means we might have to come the day before or the day after or something like that. But we will try to combine it in with the assessment activity. But of course, if you're operating 20 of these things, we're going to sample on a square root plus one basis. So until we know who's got what, we're not going to exactly know, but the discussion will be had with each authorised rep in the run up to the and planning of the assessment activity of where we want to go. If there aren't any, or for some reason they are, thousands of kilometres from the site where your static site, then we're going to have to travel to that site as an independent activity. We've got to make sure that we do our reassessments and our assessment activities and focus on what's going on in the facility. And that normally takes most of a day, or even more than that sometimes. Often an asbestos organisation has other testing going on as well, so we take a multiple team, and we need to make sure that in the situations where the sites are further away from the static site than would be practicable to go to, you might have to go to another, several hundred kilometres away or something, then we will have to plan that as a separate exercise. And those things will be chargeable in the normal way we charge. We will try not to do that. I just envisage that is probably the worst case scenario, but just because they're a long way away doesn't mean to say that on occasion we won't have to visit them. There is, I think, a sort of a summary here somewhere. Trying to scroll. The things you need to do are exactly the same things you do in any laboratory situation. You're looking at the accommodation, we're looking at the equipment, the personnel and supervision arrangements and records. I mean, those 4 dot points there. It's really a mini laboratory, isn't it? Put on the site of the activity for the convenience of the customer. There's an option here about whether or not you do this overseas. really what you should be doing is going to the accreditation body in that other economy and making sure that they're satisfied with what's going on, and they might be able to assess that for you. What else can I say? So I've described the process. Is there any hands?
No hands. Any questions? Anyone has asked a question in the chat, if you could raise your hand so I can disable your microphone, that'd be great. Thank you.
Oh, I see Linda's asked. Oh, hold on a minute, there are a few things here. What about the dedicated portable actually? So I think, okay, so David asked a question, and I think that's similar to the question that Bob asked. There are organisations that have like bands that go around. So yeah, that's quite possible. Thank you for putting on your camera, David. It's nice to see a smiling face. Looks like it's sunny over there in WA. It's not sunny here. Vina Beera said, is a field site notification form required to be submitted for every activity? Yes, you have to. Well, you have to advise us when you issue a new, when you start a new one. Yes, so you do have to tell us that. Under the field site section, it mentions sampling and/or testing as all samplings will be done on site and not in the base site. Does it mean NATA needs to be notified of all sampling being completed, even if no on-site analysis being completed? Well, you've established the field site. You've told us where it is. You operate it. We might not. Some of them might open and close before we do an assessment activity, but we will then look at the records of what went on at those field sites. So we don't need to know exactly what's going on for every minute of every day that the thing is operating, if that is the question I'm being asked. Yeah, I think that's, yes. Once you've established it and you've met the criteria to establish it, then it runs for the period that you say it's going to run for. Linda, re-existing field size, is it 12 months from the original notification to NATA or 18 months? Well, Linda, I would suggest that in order to be fair, we need to sort of apply that. We're going to have to apply the rule of 12, aren't we? I think. I'll take that one on notice because I can get very confused here. But yet, I would suggest that if they've been running for 8 or 9 months, it depends how long we're not going to know until until we know the period they're intended to run for, are we? Whether or not they're going to meet the criteria. I'll ponder that one because I do see an issue there with if you thought you had 18 months and now you've only got 12 months. But I'm going to need to know which ones have got when you establish them to know how that might apply. But I'll give you a firmer response. Read, Melanie. The question being asked is, does a field site include just sampling? No, oh, sorry. No, well, no, you can do the analysis there. I believe that you could do the phase contrast and the PLM in the field site. That's the whole point of, you collect the samples, you take it into your field site laboratory, and you do the analysis, and it's much quicker than transporting well, allegedly, it's much quicker than transporting the filters back or the samples back to the main laboratory. That, I understand, is the whole purpose of why field sites were established in the first place. I'm going to ask Luke. Luke's on the call. Is there anything else? Is there anything else I should be covering here? I think I've covered everything I wanted to cover, to be honest with you. The process. So the setup process, the notification, the way we will organise our assessment activities going forwards. This document was published, is effective in September. So, I mean, we're in July, just start of July, so you've got a couple of months. September means the whole of September. We don't sort of say the 1st of September. But as I said right at the start, the publishing date and the implementation date, they're guidance for you to get on with it. You know? Don't leave it until the week before to suddenly say, oh, now I need to notify NATA of all of this. There is no reason why you can't notify us tomorrow if you've got these things. But if you really want to hold out, then you can hold out until September, and you can send the information into our staff. I was anticipating a few more questions on this, so that's why we had a 2-hour meeting. But if
Well, raise hands if there are any more.
there's something, I think
Okay.
that's
Lancey's got a hand up. Neil, Lancey, you can go ahead and unmute.
then there's Karen's got a question.
You'll have to unmute yourself, Lancey.
Oh, thank you. Just try to unmute. Just another question going back to the volume measurement. Isn't NATA have any of the suggestions how the laboratory can assist our client to obtain the concentration if it's unendorsed report is not allowed?
Well, no, it's an issue that you'll have to deal with sort of a contract review issue between you and your customers. Nancy.
Okay. So as long as we have an agreement with our clients.
Well, you have an agreement, but the agreement has to sort of comply with the criteria I've discussed here, and including the idea that you need the requirement that you as the accredited facility, there's no way you can issue the quantitative result if you're not in control of the process, as I've described. So we've had suggested options like, well, we might unendorse the report, we might say this isn't covered by the accreditation, and we might issue another report, but you're still accredited for the test. And so you can't avoid the accreditation criteria by avoiding the endorsement.
Yeah.
I think the only way you can do it is the customer to somehow calculate it. And I'm not in favour of unless you know your customer is competent to do that calculation. I think the best thing is to consider how we might, as an industry, in this asbestos space, or in the volume measurement space of all these different, how we might come up with a better, more opportunity for sampling, accredited sampling. That to me, I think is how I think is the ultimate goal of everything. Is, you know, is there a way of the industry being accredited and being able to have more assurance of the samples that are being collected? Because across the whole of NATA, sampling is not something that's done wholeheartedly in all industries. It's a trickier thing to do because we have to go and watch it. I mean, it can be simulated during the assessment activity. There's, you know, at the site, there's no reason we can't watch you doing the pumps and ascertain that. But occasionally we're going to have to go out into the field to one of your field sites or somewhere else and watch this activity occurring, because there is a difference between seeing it in a sterile, you know, sort of normal laboratory setting than it is to go out into the field and tell how it's actually done by the operatives in the field. Now, there's a hand from Pal, but there's also a question from Karen. If the field site continues for longer than 12 months, is it required to become a permanent site? And is required to become a permanent site? Yes. Well, what happens is, Karen, that if it goes more than 12 months, then you have to enter into the application process for a normal site, and the fees apply as normally with any other application for accreditation. And I don't know, I'll go for this other question here. Thank you, Neil. Rare insight from the client perspective. When you engage a consultant who assures you they are competent, you expect them to collect reliable, high-quality information about a hazard and provide a clear interpretation of enables informed risk decisions. Clients shouldn't have to make sense of the data themselves. That's a big part of the value a competent consultant brings. Well, yes, I agree, but I think the consultant themselves, the decisions and the interpretation of the data that they make is again based on the quality of the data, isn't it? And I think any consultant who's saying, oh, well, you know, I've looked at these numbers and this is a problem or this isn't a problem, but they're not aware that the quantitative results that are generated cannot be assured as we've described today, then I think that's a bit of a risk. I mean, we are talking risk here. I mean, who wants to take the risk that one day someone challenges you and said, oh, well, how do you know that pump did suck 600 meters of air through it? You know, you can't prove it. In an emergency situation, in an emergency situation where a client requires us to establish a field site at very short notice, what is the process? So In an emergency situation, Vina, I think we're talking about the ad hoc. Remember, we talked about the ad hoc right at the start, and then there was a discussion about how long the ad hoc might be established for. That's why we made this distinction here between planning for something. You know, somebody comes to you and says, I'm going to refurbish this 10-story building. It's going to take 6 months. We're going to go floor by floor. We want you there on site to be able to give us sample return in 6 hours or whatever. That's different to the council phoning up and saying some person is, you know, building is burnt down. We think there's asbestos contamination. Can you come and have a look? That's the difference between the two things. So you wouldn't be setting up a field site as such. You'd be doing the ad hoc testing. Jesse.
Neil, Powell's had his hand up still. I'm going to allow him. Powell, go ahead, unmute yourself.
Hi, Neil. Sorry, I came to the meeting a tiny bit late. But just in terms of what I was saying before about the technical assessors being aware, so I was informed that it is anecdotal on my end. I was informed that each sampling activity had to be videotaped or a video call to supervise. So it would be nice to have at least those minimum requirements, because if you've got 100 pumps out on site, for instance, it's going to be impossible for a video call to be made 100 times a day. It's just at least having that minimum standard would be good to have.
Well, I don't know who informed of you of that, but I'm the sector manager for life science, and I'm saying that's not correct. You don't have to video them all. It's up to each individual conformity assessment body, laboratory, to determine what is appropriate supervision, depending on the nature of what's going on, how many staff are doing it, the complexity of what's happening, and how you might gather that evidence to demonstrate to NATA, the accreditation body, that you do have appropriate supervision. Now, videoing is one thing you can do, but, you know, I'm not saying watching someone continuously through a camera is not, you know, appropriate.
Yeah.
In my opinion.
And just one more, just further to that. In terms of samplers having accreditation, like a separate company having accreditation for sampling, what's the onus on the labs to make sure that accreditation is still current? Like if something was to be suspended by a separate external company, would they inform us of that? Because again, you know, if you're dealing with a few hundred samples, different clients, checking that on a daily basis or a regular basis is more difficult on that end.
So if we were to accredit an organisation for sampling, and for some reason we suspended that accreditation because they weren't meeting the accreditation criteria, for instance, then they are required to inform their customers that they've been suspended. It is also, the suspension is published on our website. So if you have a customer, if you're using an external party for sampling, you can check. You can just ask them, you know. But they're supposed to tell you. They're supposed to be upfront and tell you. And when we suspend a facility, they are sent various documents, one of which is a requirement for them to indicate they have advised their customers.
So that they would be forced to let the labs know that there is a suspension. Okay, sure.
It's an obligation of the accreditation criteria, the rules of accreditation, that if you are suspended, you advise your customers that that's happened.
Thank you.
And in certain circumstances, depending on, so we have memorandum of understanding and deeds of agreement with certain regulators, not many in the asbestos space, but in those instances, I will inform that regulator that facility X, Y, and Z has been suspended as well. Looks like some sort of AI is generated a summary. So Jesse, what about labs reporting X per unit surface area where an external sampler takes a sample and provides surface area? Should that be the same as X? Well, it's the same scenario, isn't it? If you do not have supervision over, I'm assuming you're talking about some sort of template or something on the wall. If you don't have supervision over that and you don't know that the sample was taken and is representative of whatever surface was sampled, etc., then again, I don't think you can really you can have any assurance that of the size of the template or how the template was used. You need to, if an external party is doing that, then you need to have supervision of those activities, or again, it can't be a quantitative result. That was for Jesse. Any more hands?
No hands at the moment.
Okay, well, I hope that was useful. I went on for an hour and a bit, about an hour actually, because we took a few minutes. Oh, Cassandra Matters has a question.
Go ahead, Cassandra.
Just saying, I think Jesse's question was about the tape sampling for mould, because they use a piece of adhesive tape to sample.
Oh, yes.
For that. I think that's what that question was related, not so much a template. So maybe if you could provide,
I've
like,
allowed
would the tape
Jesse's
sampling for mould fall under the volume measurement as well?
Well, it's not air going through a pump, is it? So
Yeah.
It's not the same thing. So yeah.
I've allowed Jesse's mic so she can unmute and confirm that if she likes.
Sorry,
no, it was more for something where you might take, a sample of illicit drug residue off the surface or something like that from a, templated 10 by 10, as Neil was saying, and you would sort of provide that 10 by 10 or the surface areas of the lab, and they would report the sort of per unit concentration. Just wanting to make sure that, and more broadly, just generally whether it applies to everything where sort of some unit or measurement is provided by a sampler and they're not sort of part of the controlled facility.
Anywhere where you are using a party other than the party under your direct supervision, there's a risk. They're not. Do you know they're doing it? It's on your reconnaissance. You're on your own reconnaissance to go and check that. And that's what I saw at Lindsay Neal. Oh yes, Lindsay.
Yeah, Lindsay, go ahead. You have to unmute yourself, Lindsay.
Thanks, Anne-Marie. For Jesse's comment there about sampling for a surface for area. You mentioned sampling for illicit drugs. So probably one of the methods you might be thinking about is one of those NIOSH methods out of America. If you look at those methods, what they actually require you to do, if you're doing the sampling properly, is you're actually meant to provide the lab with two samples. One's meant to be a first go at basically recovering all the analytes on that surface. And then the second is meant to be a go over that surface that's meant to indicate you're now collecting less than 10% of the analyte on the first swap. So there's actually an indication there that you've actually recovered all the analyte from that surface.
Well, that's the other thing, isn't it? There's one thing to swab the area. There's another thing to make sure your swabbing is actually lifted off what you want, especially when you're talking about sort of minute quantities of illicit substances, which isn't quite in my area, but I'm happy to discuss it today because I mean it's in our legal and clinical area, but the same thing applies. So if that's it, I can give you a bit more of your day back. Thank you all. If you can think of, if a question arises, then you can always email me. But I think we've covered all we had to cover today, and let's take the next step forward in all of this and see how we go. And I look forward to hearing that we've received a few forms and notifications of field sites in operation. Thank you very much.